Hello everyone. Today we are going to discuss about the different types of media which we used in cell culture. Natural media, synthetic media, the composition and the preparation. I am Dr. Priya Josan Akkara from the Department of Life Sciences, Christu Jayanti College Autonomous, Bangalore. Now, what is the essentiality of a cell culture media? What does it refer to? So, we know that cell culture is a technique where cells are allowed to grow under controlled conditions. So, we take out the cells, we make a primary explant, we take a primary explant, from there we grow a primary culture and we grow the cells under controlled conditions. So, animal cell culture is a technique in which the cells are removed from the tissue and are allowed to grow in a favorable artificial environment. Cell culture medium is an essential component in ex vivo cell maintenance because that is like giving life to our cells. Basic media provide all the necessary elements for growth and survival. So culture media can be either of natural origin, they are the biological fluids, or it can be synthetic. So we are going to discuss about the different cell culture medium. So the media composition, so what does the media contain? It contains the sources of energy like glucose, fructose, nitrogen sources like the amino acids, vitamins, the water-soluble vitamins, vitamins B and C, inorganic salts, sodium, potassium, calcium, magnesium, fat and fat-soluble components, fatty acids, cholesterol, antibiotics, growth factors and hormones, oxygen and carbon dioxide concentration. So all these are available in the cells in our system, but we are not realizing how well God has created us with all these factors. So when we are trying to create an artificial environment, we realize that there are so many components which are required for the cell, and these are the ones which we are adding into the media. So other than that, the physical environment is also maintained. For example, the optimum pH is maintained, the temperature, the osmolality and the gaseous environment is maintained. And along with the media, most of the times we also add serum because it contains a lot of nutrients. Now in animal tissue culture, two types of culture media are used. They are the natural media and the synthetic media. The type of the medium relies on the type of cells to be cultured and its objective. So depending upon what type of a research work you are doing or what type of cell lines you are growing, depending upon that you can decide whether you have to use a natural medium or a synthetic medium. Now what are these natural medium? Natural media can be plasma, it can be serum, tissue extracts, clots, and plasma clots or plasma is most commercially found in the liquid state and can be prepared in the laboratory. Then we have other biological fluids. Okay, for example, it can be a amniotic fluid, it can be a pleural and ascitic fluids, hemolymph for insects. Suppose you're growing insect cells or aqueous humor from eye, and of course serum, of which the serum is the most popular or most commonly used natural media. So serum is a plasma after the removal of fibrinogen. You all must be clear with what is plasma and what is serum. Serum is the plasma after removal of the fibrinogen. So it can be with or without other nutritive substances, may be used as the entire culture media. So serum itself is so rich in nutrients that they can help the cells to grow. Other than that, the third one is the tissue extracts. So out of this tissue extract, the most common one which is used is the chick embryo extract. And bovine embryo extract can also be used. Spleen, liver, bone marrow, leukocytes can also be used. And substitution for tissue extract can be a mixture of amino acids and certain other organic compounds. Of course, we have substitutes for all these. So these are the major commonly used tissue extracts. Coming to synthetic media, so we just discussed the natural media. Natural media can comprise of either plasma or a serum or a clot or a tissue extract. And tissue extract can be of different types and biological fluids can be from different types. Okay. Now coming to synthetic media. So synthetic media are made up of partly or fully defined components and they are usually formulated. And it contains amino acids, salts, vitamins, hormones, growth factors and carbohydrates, and it can be used to create more specialized media. Depending upon what you are working with, your media can be specialized by addition of serum. And continuing with the synthetic media, various artificial media has been employed. They help. What is the advantage of using synthetic media? They help in the immediate survival. It is a balanced salt solution with specific pH and inadequate and adequate osmotic pressure. It helps the prolonged survival of your cells and indefinite growth, and it also helps in specialized function. Now coming to synthetic media, as we discussed about natural media, synthetic media also can be classified. So let us revise. Natural media we discussed, which are they? They are the plasma, the serum, the clot. Of course we have the biological fluids, and of course we have the tissue. For example, we were discussing in the previous slides. Now coming to the synthetic media, we have different types. They are the serum containing media, serum free media, protein free media, and chemically defined media. So there are 4 different types of synthetic media. Now let us discuss them one by one. Serum containing media, of course we usually use a serum, and most commonly uses your FBS or fetal bovine serum. That is the most common supplement, and it is employed as an economical supplement. It's not a very costly one, and it supplies water insoluble nutrients, protease inhibitors, hormones, growth factors. Okay, so all these are helpful. Serum free media is being utilized nowadays, because if you are working, or if your research work is based on immunological markers and things like that, it is better not to use a serum, because that will interfere with your studies. So in such cases, you use serum-free media. Presence of serum in media can result to serious misinterpretation of your results. So depending upon what you are studying, accordingly you can use either serum-containing media or serum-free media. They are specifically used to designed to promote the culture of a single type of cell, and the components are referred to as a defined culture media. Then this is very common nowadays, the chemically defined media. So depending upon what you require, okay, accordingly you add the factors. So it contains ultra pure inorganic and organic ingredients which are free of contaminants and contain pure protein additives such as growth factor. So chemically defined media contains the protein additives and growth factors, and constituents are produced by genetic modification in bacteria or yeast with the addition of vitamins, cholesterol, particular amino acids, and fatty acids. So chemically defined media is totally different from your serum free media. Now last one is your protein free media, devoid of any protein, because suppose you want to study for example protein expression studies. In that case, it is always preferable to use protein-free media. Otherwise, the protein in the media itself will deviate from you. You will get results which are not easily. You will not get the right results. This protein will interfere with your protein expression, whichever you are targeting that particular protein. So usage of protein media enhances superior cell growth and protein expression in contrast to serum supplemented media, and enables downstream purification of any expressed product. So this is also another important thing. So it is easier to purify. Downstream processing is much more easier if you are going for protein free media. So that is the main advantage of protein free media. You don't have to spend much on the downstream processing part. Now coming to the preparation of media. So let us take 2 important medias which are the most popular medias. DMEM, okay, the first one, Dalbecos modified Eagle media. Suppose you want to prepare 1 liter. This also comes as a readily constituted one as a liquid medium, or you get to buy the powder. So if you are buying the powder, you have to prepare 900 ml of distilled water. It is usually better to go for WF5 water. Add the 1 liter powder, 13.3 grams to the water. Always refer to the bottle. Check in the water. In the bottle, what is written. Accordingly, you weigh and add 3.7 grams of sodium bicarbonate or 49.4 ml of 7.5% sodium bicarbonate solution. So usually sodium bicarbonate will not be there in the powder, which we have to add at the time of preparation, and you have to adjust the pH 7.0 to 7.4. Usually the indicator inside the media will help you to understand whether the media is having that particular pH. So when the pH changes, the color of the medium also changes. So if it is going towards yellow when the cells are growing, which means the media is becoming more acidic. So add distilled water up to 1 liter. Now the media is ready. Next step is you will have to filter the media. Okay, now we were discussing about DMEM. GMEM is another popular media. Here also, as per the whatever it is written in the bottle, suspend 15.5 grams in 900 ml of culture grade water with constant gentle stirring until the powder is completely dissolved. Never ever heat it No, never ever heat the water or the medium, okay, because it contains lot of proteins which will get denatured. You add 2.75 grams of sodium bicarbonate powder, adjust the pH and make up the final volume to 1000 ml. And sterilization is important. Always remember, media used for animal cell culture can never be autoclaved. It should be filtered and sterilized. Sterilization is used by filtration. So sterilize a medium immediately by filtering through a sterile membrane. It can be a syringe filtration if you are doing only a very small amount or volume of media. If it's a larger volume, you can go for other types of filtration techniques with a porosity. Usually you use a filter of 0.22 micron size using a positive pressure rather than the vacuum, because it might minimize the loss of carbon dioxide. And aseptically add sterile supplements. So all this you have to do inside the biosafety cabinet, and then you can add sterile supplements as required and dispense the desired amount of sterile medium into sterile containers. So all these containers also should have been sterilized in the beginning, and after preparation of media, you can actually aliquot your media into the containers and keep. And at the day of whenever you're growing the cells, you can actually add the serum into it, 10% FPS, you can make the media, and then you can actually add it to the cells to grow. So usually it is store the liquid medium at 2 to 8 degrees Celsius and in the dark till use. Always open the media only inside the biosafety cabinet. So these are the other medias which are used in the animal cell culture, the PBS. Of course, FBS is used as a supplement or as a, for the growth, the serum, and RPMI media is a usually used media. And these are the references. So to conclude, we were discussing about the different types of media, the natural media, the synthetic media, and in natural media we were discussing about the serum, plasma, clot, and tissue and extracts. And then in the synthetic media we were discussing about the serum free media, serum containing media, protein free media, chemically defined media. And then we also discussed about how to prepare media and how to sterilize media using filtration. Thank you.